Hi, my name is Rodrigo Villalobos,
And my name is Armando Alvarez.
and our project
Is
honor science seminar is bioinformatics.
on the HIV subclass AE, subtype AE.
Our first article, in which we first found our background research, was a TED Talk by Ella Saldaña. He is a renowned researcher, and he discusses in this TED Talk the new resurgence of a brand new subtype called subtype AE, which is a way more aggressive subtype than the subtypes previously mentioned in previously known for the rest of the disease, the virus. Subtype AE, unlike subtype B, is more aggressive and is known to have some sort of resurgence and act as a have some sort of resistance. I mean, in contrast to subtype B, which can be treated and diminish its effects, so preventing it from leading to AIDS.
The second article is one HIV strain in Philippines must be better researched if rates of infection not to explode, scientists warn. What happened in this article, it talked about how there was a potential of a new epidemic of a new subtype AE, and it talked about how it's more aggressive than any other HIV strain, and it's more resistant to antiretroviral drugs, and it leads to AIDS faster than every other subtype already found.
So statistically speaking, subtype AE is more prone to lead to AIDS than any other subtype known. And currently, subtype AE has encroached upon various first world countries' population, not just the Philippines, not just third world countries, but countries like the US, even Canada and Australia.
It mostly affects young, sicker teens with high resistance to antiretroviral drugs.
Therefore, the patients who suffer from subtype AE HIV are known to die quicker and lead to AIDS quicker than those without.
And as you could see in the stats, UN claims that the prevalence of HIV shot up by 140%, and that there was a total of 56,000 cases in 10 years, and only 10,000 new ones in 2016 alone. This third article is Study Examines Transmitted HIV Drug Resistance in Asia. This article talked about they were testing, there was 3 testing sites, Malaysia, Thailand, and Hong Kong between 2007 and 2009 with 682 patients. And they gave us statistics of how they got the virus and what types of not immunities, but resistances they had
to anti-viral drugs.
Mm-hmm. And there was a editor, an author. He talked about how that we need a better understanding of the prevalence of the transmitted resistance. If not, all information of HIV would be gone to waste with this brand new HIV virus subtype AE.
Our testable question is, how can we use BLAST, which is an algorithm tool that allows you to compare the similarities in each gene or codon to study the relationships of HIV transcriptions that result in mutated or more aggressive HIV subtypes. Our problem statement was that me and my partner don't think that in a world with ample amount of resources, it is fair that we deprive from having some sort of knowledge of increasing the quality of life of those suffering from this virus.
The motivation for this project, we talked about how HIV and its implications have caused patients not only physical harm and despair, but also enabled scrutinization and ostracization of these victims.
Victims, even here in the U.S., have faced ignominy throughout the beginning of this virus, it is known that in previous years or previous administrations, they have even gone to such lengths as prevent citizens with HIV from entering the country. So that's just another way of discrimination.
This is our design diagram. We were planning to use the control group, the HIV subtype B, which is the most common subtype, but only accounts for 12% of the entire HIV virus community.
Yeah.
We're using HIV subtype 8E, which is the new, more aggressive subtype. HIV subtype C, which falls under the same family as HIV subtype B, which is HIV 1. And then subtype HIV subtype CRF19, which was founded in Cuba and are talking about it as a more aggressive than AE, but we do not know. Yeah.
There is not enough statistics. But even to enlighten you guys, HIV, different subtypes and its different mutations and just transcriptions are so different that it is equivalent to a human having, or
let's say an ape having a baby. The baby is a gorilla. The gorilla has a baby, and the gorilla is an orangutan.
Yeah.
A orangutan has a baby, and it's a chimpanzee. That's exactly how it is with HIV. Every time it enters a new cell, it talked about how the DNA sequences vary by 35%, and with humans, the genetic differences is only 0.1%. Between humans and apes is 5%, but with HIV cells, the mother and daughter cells is 35% every time it enters a new cell. So it's not counting generations.